This fly is walking with the wiring diagram of a worm. Researchers connected the C. elegans connectome to a physics-based fruit fly body, trained the interface, and produced a realistic six-legged gait. They built this digital chimera to challenge a larger claim. Earlier that March, Eon Systems showed a virtual fly walking, grooming, and feeding, controlled by a model derived from the mapped fly brain. The company called it an embodied brain upload. Both systems move. The hard question is what that movement actually proves. The foundation is extraordinary. In 2024, FlyWire published a wiring diagram reconstructed from one fixed adult female brain, 139,255 neurons, and 54.5 million chemical synaptic links. That is not a living brain. It does not capture electrical junctions, neuromodulation, plasticity, memory, or the internal state of a living fly. But it is detailed enough to generate testable predictions. A model built from this map treated neurons as leaky electrical units and synapse counts as weights. In one feeding circuit screen, 10 of 11 predicted positive cell types triggered the expected response in live flies. Only 4 of 95 predicted negatives did. That is a real scientific win. Wiring used to predict an experiment. Eon connected that model to NeuroMech Fly, an anatomical body inside the MuJoCo physics engine. Sensory events activate chosen pathways. Brain activity updates. Selected descending neurons become commands for turning, forward motion, grooming, or feeding. Movement changes the next sensory input. The loop closes every 15 milliseconds. That integration creates a useful testbed for linking neural hypotheses to behavior. But Eon's own technical account draws a boundary. Walking uses modified controllers already trained to imitate fly movement. A small set of descending signals acts as control handles. Several brain-to-body mappings were chosen by hand. The full motor hierarchy is absent. Current vision barely affects behavior, and internal dynamics lack biological validation. The Connectome did not simply wake up and discover how to walk. Then came Digital Sphinx. The team randomly projected 148 body sensors into 82 worm sensory neurons. A reinforcement-learned decoder mapped worm network activity into 42 actuators controlling 6 fly legs. The result reproduced convincing joint motion and coordination. Biologically, it is deliberately wrong. Worms do not have legs. The neural identities have no meaningful relationship to fly locomotion. Yet a flexible, learned interface can still make the body look right. Digital Sphinx does not invalidate every component of Eon's system. It does not directly test Eon's fly-derived connectome, neuron model, vision system, body physics, or closed-loop integration. It uses a different architecture and a trained decoder. What it challenges is behavioral validation. If a worm network can pass the walking test, realistic walking alone cannot prove the simulated biology is right. A 2026 connectome makes the interface even more important. BANC mapped an adult fly's brain and nerve cord together. Its analysis suggests that many effectors receive their strongest estimated influence from local sensors in the same body part, while the brain coordinates and supervises those modules. Behavior is distributed across brain, cord, sensors, muscles, and environment. BANC's influence scores are structural predictions, not recordings. But the result shows why the bridge between brain and body is not plumbing. It is part of the computation. A convincing upload needs harder tests. It should predict unseen neural activity, respond to lesions like a living fly, preserve known internal dynamics, and beat rewired random or cross-species controls without hiding the task inside a black box interface. So, did scientists upload a fly brain? They built a remarkable closed-loop research platform from a landmark wiring diagram. They did not recreate a fly mind or prove that wiring alone explains behavior. Digital Sphinx does not erase Eon's achievement. It raises the standard for interpreting it. A body that looks right can conceal a brain model that is wrong. The next breakthrough is not a prettier walk. It is a model that makes risky biological predictions, then survives when real flies are asked for the answer.